Inside a complex organism, sending a signal from the brain to a peripheral muscle is a massive logistical challenge. A standard spherical cell simply cannot bridge that gap. Chemical diffusion is too slow, and undirected secretion over distances that span meters is physically impossible. To solve this, the nervous system relies on a highly specialized piece of biological hardware, the neuron. This single cellular unit must execute 3 distinct tasks in rapid succession. It has to receive incoming chemical data, conduct an electrical signal across a vast physical distance, and then transmit that signal flawlessly to the next target. To achieve this directional, high-speed communication, the cell has to abandon a standard shape entirely. It physically segregates its anatomy into dedicated, compartmentalized operational zones. These zones consist of a central metabolic engine, an expansive receiving network, and a single dedicated output wire. This diagram outlines the target architectural layout of a fully developed multipolar neuron. We can see all 3 specialized zones laid out in sequence, from the input channels on the left to the output terminals on the right. The neuron is a highly engineered biological circuit, where morphology is dictated entirely by functional necessity. We begin structural reconstruction at the core of the cell. This irregular, star-shaped center is the cell body, also known anatomically as the cyton or perikaryon. Inside, the cytoplasm, specifically called the neuroplasm, contains a large central nucleus with a prominent nucleolus. Surrounding it are standard organelles, mitochondria for energy production, neurofibrils for structural support, and the Golgi apparatus. But the defining feature of the neuroplasm is a dense concentration of rough endoplasmic reticulum clustered tightly around the nucleus, known as Nissl bodies. These heavy industrial factories synthesize the crucial proteins and neurotransmitters required to make the system run. Branching outward from this metabolic center are multiple short processes that form the cell's input network. These are the dendrites. They operate as the neuron's non-myelinated receiving antennas, capturing incoming signals from neighboring cells. If we examine the internal texture of these branches, we find a critical diagnostic marker. They contain the exact same Nissl substance found inside the cell body. Because they share this internal chemistry and contain Nissl bodies, dendrites are essentially metabolic extensions of the soma itself, stretched outward and optimized solely to collect incoming data. On the opposite side of the cell body lies a funnel-like region called the axon hillock. From here, a completely different type of anatomical structure begins to emerge. This is the axon. It is a solitary, exceptionally long outward process built specifically for direct signal conduction. There is a strict physiological boundary at this junction. If we look closely at the axoplasm, the specialized cytoplasm inside the axon, we see that it contains 0 Nissl bodies. Without its own manufacturing centers, the entire length of the axon is completely metabolically dependent on the distant cell body for its protein supply. This dependent axoplasm is firmly encased within its own dedicated plasma membrane, known as the axolemma. Running a bare biological wire over a massive internal distance introduces a serious problem: rapid electrical signal degradation. This technical diagram details the biological solution. Looking at the right side of the structure, we see the myelin sheath, a vital layer of insulation present in most peripheral nerves. This sheath isn't part of the axon itself. It is actively formed by independent support cells called Schwann cells, which wrap themselves tightly around the conductive wire. The outermost living layer creates a protective outer covering known as the neurolemma. Notice that this insulation is not a single continuous tube. It is periodically interrupted by tiny exposed gaps labeled here as the nodes of Ranvier. The resulting structure is an isolated, heavily insulated output wire. It completely trades local metabolic independence for sheer conductive speed and range. Following this insulated axon to its absolute physical endpoint, the signal wire finally diverges into multiple terminal branches. These terminal branches are called telodendria. Each individual telodendrion ends in a small, specialized swelling known as a synaptic knob. Inside these distant knobs lie the precise chemical packets, the neurotransmitters, that were originally manufactured by the Nissl bodies all the way back in the cell body. This brings us to the critical interface point where these knobs meet the dendrites of the next target cell. This junction is the synapse, allowing the nerve impulse to chemically jump the gap and continue. The entire elaborate architecture of the cell, the high-capacity metabolic engine, and the rapid transit insulated wire exists solely to deliver neurotransmitters across this tiny synaptic divide. With the full anatomical model assembled, we can classify neurons based exactly on how many processes extend directly from the central soma. The specific model we just constructed, featuring numerous dendrites and one single axon, is a multipolar neuron. This is the standard layout utilized by motor neurons. This comparative chart displays 3 distinct neuron shapes. Alternate sensory tasks require different morphological configurations. The bipolar configuration, possessing just one dendrite and one axon extending from opposite ends, is highly specialized. It is found primarily in the retina and the olfactory epithelium. Then we have the unipolar, or pseudo-unipolar layout. Here, a single process exits the soma before splitting. This structure is heavily utilized in the dorsal root ganglion. Because function is so strictly bound to shape, physical damage to this architecture translates immediately to clinical failure. If injury physically severs the axon wire itself, the clinical result is a total loss of electrical conduction along that pathway. The signal stops dead. But damage can also target the insulation rather than the wire itself. This process is known as demyelination. Demyelination strips away the myelin sheath without initially breaking the axon. The result is a severe decrease in conduction velocity, causing precisely timed electrical impulses to stall, leak, or short-circuit entirely. In neurobiology, strict anatomical segregation is what dictates biological function. Maintaining that structural integrity is what dictates clinical health.